The first thing that I'm going to talk about is the component that we call the otoscopy. Thank you, Mac. Otoscopy. Otoscopy is a part of what we call the trusted stack. This is a solution that we actually shipped to a vendor of secure digital control devices. These devices protect the power grid, and these are protective relays and energy management servers. What that component does is it sits in the kernel. It examines the kernel environment on certain events, such as system calls, and other uses of the kernel internal interfaces, because of course, even though you see 3 layers here, the kernel itself breaks into a whole bunch of layers, VFS being the most famous of them, but usually your typical system call is dispatched through at least 3 layers of indirection. And we can catch and examine the state of the kernel at any one of those interfaces. And it turns out that it's possible to do that with good enough accuracy and fast enough. So in fact, we have this not just for servers, but also for embedded devices. And some of this even includes disassembly of the kernel on the fly. So this, of course, latency requirement in protection relays are very, very heavy. And it has taken us quite a bit of working with that particular vendor to get this to work. But they are now shipping this as a part of their product. And what you see on top of that is a system that spans all 3 layers of an application. We call it ELF-back. It enforces ACLs between different components of the ELF runtime, which is to say all of the Linux runtime. And it's an integrated solution, which we again plan to transfer to a customer sometime this year. So the other word that you see on the screen, the crossing guard, is a solution that protects the kernel from the bottom, from bus side crafted inputs. This is a vast attack surface. It is completely unprotected. It is, as far as I know, completely unprotected in all sorts of embedded devices, including control, including medical. And we discovered that and we explored that with the phase dancer device, which can fake any USB device. So what do you do in front of an input that is crafted and malicious on a network? You put a firewall in place that checks all of those different data structures that can be crafted. Of course, it's best to fix the code, but yeah, good luck with that. So we, again, look at the USB stack as an entire collection of interfaces. We hook those interfaces and we model the data types that cross them. This is a thesis that is close to completion, and a prototype exists. So this is kind of like the net filter or packet filter, or BPF, if you will, for USB. And it models the data, the very many data descriptors, the kinds of data descriptors that can make a device appear to be a mouse and a disk at the same time. We had just such a paper in Wood. I invite you to have a look at that. So we model all that data, and we model the context. And Peter Johnson likes Haskell, so he did that modeling work in Haskell. From that Haskell, C code is generated, and evaluation engines to run the firewall are produced. And that's, we hope to produce a full-service USB firewall to those systems that have exposed bus interfaces and need that kind of protection. Thank you. Questions?
We don't right, then I'll bolt to my own workshop.
But I do think it has. I do think it has real utility in the world of medical devices. So.
Our primary focus was call systems, but we're looking at some extending. Thank you. Sorry for the red.
So I'm just going to channel Carl Landwehr since he wasn't able to come. He's proposing or planning a workshop this fall on, I guess I should resize my window here for you. A workshop on building code for building code. So he's, some of you might have heard his lecture or seen his paper on the notion of the need for a building code for software, much like we have building codes for structures like hotels and homes and so forth, essentially an engineering code, if you will, for software. And so he's proposing a workshop on a building code for medical device software security and safety. And that's going to be in New Orleans in late November. There's, let's see. Sorry. Somewhere in here there's a call for, here we are. So he's looking for participation, especially for people from industry, especially from people from the medical device industry or the medical software industry, experts in software engineering, et cetera. And so this is an NSF-sponsored workshop by invitation only, but you can arrange to be invited by sending him some email with some information about why you want to come. You should read this page, and I'll give you the URL again in a moment. But here is a note of not more than 600 words and sort of what you want to discuss at the workshop. And here's the address, BC as in building code for MDSS for medical device software safety at gmail.com. And the URL is kind of longish, but if you were interested in this meeting and would like to attend, just shoot him an email. This is Carl Landwehr. Many of you know him from NSF and IARPA and places like that. There is funding available for travel. Again, this workshop is funded by the NSF, so if you're interested in going and don't have an appropriate funding source, you can possibly get some support from Carl from the NSF. Okay?
So I think Eugene ran out thinking that the last speaker had taken his dongle, but maybe this is it. So maybe it's actually here, so he's going to probably be a while. So I just wanted to make a couple of closing remarks. You're all going to receive, if you registered for this workshop, a survey. And if you don't think that we look at those surveys and take them seriously, just look at the way that this event has evolved. We started out with a workshop, and then we changed the name from HealthTech to HealthTech, and then we had a summit. So we really do listen. It's here. And thank you very much for coming and making this great. We have one more Rump session talk.
I'll be quick.
Which will be quick. And I'll let Eugene also thank you guys and do his wrap-up, but that's my wrap-up. So thanks for coming.
I can do my wrap-up first. Thank you, guys. So this is a representation of something I talked about at PETS, I think 1 or 2 years ago, and was co-developed with Ian Goldberg, who is not in this room, and therefore I don't have his permission. So if anything goes wrong, it's entirely his fault. Just a quick note on privacy-preserving genomic sequencing. This is a huge problem because we leak a lot of our genome every day, right? Skin flakes and so forth, and sequencing is cheap, getting easier, getting much, much faster. There's genuine concern about this. This was an article which was timely at the time I first gave this talk. You can't really hide your genes. You shed them everywhere, skin cells, hair, so forth. They can be analyzed offline without your consent. We can understand these are problems. How do we prevent or at least minimize the vulnerability, perhaps the window vulnerability? So first, a little bit of background. You've gotten a lot of this already before in the microbiome discussion. But first, let's talk about the way we get from DNA to DNA. This is the process of DNA replication. This is orthogonal, so I won't talk too much about that. But this is the part we're concerned about, that is DNA sequencing. You replicate DNA and then you look at the results. Let's talk about the way you convert DNA to proteins. So first you have to go through RNA stage and then you go through proteins. Into proteins. These images are from Wikipedia, so thank you, Wikipedia. Basically, you have a template strand, you have a coding strand, you have a gene in the middle, you have a protein complex which attaches and sort of zips along and unzips the two strands like a zipper, makes a copy of one, and then attaches a second one. Now that this is RNA, now we go to a protein, and for this we need a ribosome. Here we have messenger RNA, which was this component being created. And if I'm a bit rusty, it's because I haven't practiced this talk in 2 years. Then you have incoming transfer RNA, which is attached to peptides here, and there's a growing peptide chain which forms your protein. So the protein is what you need to make your cells work. DNA is what we want to protect. By the way, here's a much longer, nicer image of this entire complex working. Here's the messenger RNA coming through. Here's the transfer RNA, and there's the growing protein coming out of this protein complex. So there's a key idea. We can use a known technique to secure this process, and that is the introduction of kRNA. Let's just use a stream cipher and apply this to our DNA. There are, of course, some practical considerations, but we'll talk about those in the next 2 slides. So let's encrypt the DNA and decrypt it at translation time. So we'll take a stream cipher. We'll implement the stream cipher in the spliceosome by taking an extra strand of DNA, that being the keying component, kRNA. So here you see the picture you saw before. This should look familiar. Now you have the somewhat modified spliceosome. So splicing is something you do to RNA between the time it gets converted from DNA to RNA and then to a protein. First, you have to splice the RNA. So why not have encrypted RNA, which is decrypted when it is spliced? So here we have the mRNA as before. Here we introduce a new strand, which is the kRNA, or the key to our stream cipher. And here we have the pre-spliced but already decrypted mRNA, whereas this is the encrypted mRNA. So there are a number of potential benefits. I hope you see that. There's only one point of modification in practice to the running system, that is the spliceosome. This is all incrementally deployable. I can do that to myself before anyone else has to do it to anyone else. The benefits are entirely mine when I do it, so incentives are very well aligned. DNA replication itself is unaffected. You replicate encrypted DNA just like you replicate non-encrypted DNA. No problems there whatsoever. You can make this non-malleable, and you can protect yourself from potentially dangerous mutations. Just restore from backup when you see some kind of problem with authentication. So authenticated DNA, which is great because you don't have to worry about any of that unauthorized gene therapy, which is performed at you at night by NSA agents, I'm told. There is, of course, some future work. There are non-trivial considerations here. We do not have permissions to do human trials for this, by the way. So that's later. But we need some kind of way to do just-in-time password entry, or just some kind of credentialing, and including auto logout, because right now they're just totally unsolved. And we need a trusted path from whatever credential entry mechanism we have to the spliceosome here to avoid the dreaded protein in the middle attack. Another problem is preventing user death, because it's not quite as simple as I presented before. I mean, there are all sorts of things. Biology is full of exceptions, RNA splicing, cleavage, polygynylation, so forth. There are a lot of things that go on which I glossed over completely. DNA promoter processing becomes difficult because the promoter is meant to be processed unencrypted. If we have an encrypted promoter, what happens? We don't know. Password expiration and DNA degradation is another problem. We need some kind of cold boot attack resistance because you could freeze my cell, extract kRNA, you have my keying material. This is clearly a problem. And we need some kind of way to do credential delegations because otherwise you wouldn't really be able to do personalized medicine, and reproduction would be extremely hard. And that's the entirety of the talk. I can't take questions, but I welcome any comments offline, and I look forward to human trials of this particular system.